Okay, I ran into an issue yesterday when I was recording. You may have wondered why all of a sudden it just stopped. So I'm just going to pick up just a couple things out of section 7.1. I ended up leaving off talking about BPG, bisphosphoglycerate. Now, erythrocytes don't have nuclei and they don't make proteins. So essentially, the hemoglobin that they have in there, that's all that red blood cell is going to get. And so in your body, if you need additional oxygen, it takes a while to create those new and additional red blood cells. So what BPG can do is actually respond much quicker. It's a small molecule and it can be released to help us adapt to lower oxygen levels. And so within a day or two, you can actually start to acclimate. If you stay at high elevations after weeks or a month, you'll notice that it's much easier to breathe. But BPG allows us to acclimate much quicker, make small adjustments to the release of oxygen. And this is referred to as allostery. We'll talk more about this when we get into enzymes, but right now, allostery is the binding of a ligand at one site that affects the binding at another. So you bind to a protein at a site distant from the active site, and that binding influences the binding of the substrate. So it can either increase or decrease affinity. So generally, allosteric interactions require multiple subunits of an oligomeric protein. And there's ways to try to explain this allosterism. The effect of one subunit on the other. Like I said, we'll pick up a lot of this when we actually get into specific enzymes. But to help explain the oxygen binding and how it converts from the T state to the R state, there are a couple of different models. And this model here is referred to as the symmetry model, where all the subunits are going to be of the same type. Either they're all in T state or they're all in R state. And then these states are in equilibrium between each other. And so you can see the different possibilities that you could have. You could have substrate bind, and then that substrate causes everything to convert to the R state. Or it could be that, I get a second substrate to bind, and a third one, and maybe even a fourth one before I get there. Or it could be that some molecules, when they get two, they switch here. Essentially, in this idea, it's kind of an all or nothing thing. So the ligand can bind to a subunit in either conformation there, and then the conformation change alters the affinity of the ligand. So this model right here could be used to account for oxygen binding to hemoglobin, but in some cases we see a negative cooperativity where binding of a ligand makes it more difficult for the next ligand to bind. So there is a second model, and that is the induced fit or the sequential model, and that is where once the substrate binds, it's going to have a kind of localized effect on the subunits adjacent to us. And that's going to make it easier or harder, but in the case of hemoglobin, make it easier for the second one to bind. And once that second one binds, it's going to have an impact on the adjacent ones, and then that makes that easier for the substrate to bind. And so this is what's referred to as the induced fit or the sequential. So every time you add one, it allows the next one to be more easily added. And this requires strong mechanical coupling between subunits, and it could lead to simultaneous conformation changes like we see in hemoglobin. So in the example of hemoglobin, what would happen here is once the substrate bound, then all the subunits here would move to the new relaxed state. So in hemoglobin, you see that those changes are very cooperative. So either they're all in the tense state or they're all in the relaxed state. Okay, so a little other side note is there's over 1,000 variants of hemoglobins from humans that we know. So from human to human, there might be a slight variation between you and your neighbor's hemoglobin. And more than 90% of these variations just result from a single amino acid substitution. So about 5% of the people have one of these 1,000 variants. So if you know 100 people, 5 of them should have some type of variant. And so how they name these is the place where they were discovered. So such as Hammersmith in England or Bristol. How they indicate it is the amino acid that was there. This is the site. So this is actually the chain and position. In parentheses is the site, and then you got the subunit that it's in, and then what's the new amino acid. So in essence, this one changes phenylalanine to serine, and it actually weakens the heme binding. If you remember, we talked about heme is bound by hydrophobic interactions. Same with the valine to aspartic acid. Aspartate weakens the heme binding. This leucine 19 to proline, like we know, proline actually disrupts alpha helices. Glycine to valine disrupts the the B to E helix interface. Anyway, so you can see the other ones there. So some of these actually favor the meth hemoglobin, which is actually the iron 3 oxidation state, and that's really bad because you kind of remove that hemoglobin from function once you oxidize it from iron 2 to iron 3. Sometimes you result in unstable hemoglobins, and so those are degraded, and this could lead to hemolytic anemia. And the reason for that is you break down the hemoglobin, the degradation products can cause your erythrocytes to lyse or to break open. And of course, that would lead to compromising the O2 delivery to the tissues. Polycythemia is when your hemoglobin has a stronger binding to O2. And cyanosis, like I said, is those cases where the iron gets oxidized to iron 3 there. In this case, cyanosis, you're not as efficient at carrying oxygen, and so people with cyanosis tend to have a blue color, and polycythemia, you've got stronger O2 binding, they tend to have a ruddy or a rosy complexion. One of the best known mutations in hemoglobin causes what's known as sickle cell anemia. And it is caused by a mutation. And what it does is you have a glutamate that is very polar, and it's mutated to a valine, which is very non-polar. And so it just happens that on one hemoglobin molecule, you can see that you got this hydrophobic pocket. And normally nothing binds there. But if you get this modification on hemoglobin to valine, so you got this mutation, it just happens that that valine is pointing out at the surface, and you have this surface hydrophobic pocket. And so that valine can just nicely nestle into that pocket there. And in doing so, you end up with this repeating binding pattern just from that single amino acid. And so your hemoglobin ends up forming fibers. Now, this mutation is found in about 25% of Black Africans and 10% of African Americans. So pretty high abundance of this. And what happens is it causes sickling of the cell, or the cell to take kind of a crescent moon shape. And the reason for that is the hemoglobin. So here we've got the normal hemoglobin, and this is just a way to illustrate it. So there's a mutation, And that mutation essentially exposes a hydrophobic pocket and a hydrophobic residue that can link together to form some strands, and then those strands can actually form fibers. Here is a picture of a lysed red blood cell. So it had so many fibers in there that it ended up poking through the red blood cell, and you can see those fibers. You can see that this red blood cell is just full of fibers. So the reason that you get sickling is that you're packing these fibers in there, and those fibers end up poking out kind of the ends and cause the red blood cell to kind of take a sickle or a linear shape. And that's bad because now they're going to get caught up in capillaries, and that leads to other issues as you start to block your capillaries because these sickled cells are getting hindered as they're delivered through your body. There is one advantage to having this sickle cell gene, and that is that you have some malaria resistance. So if you're heterozygous, meaning that you're producing some normal hemoglobin and then some hemoglobin S, which is the mutation, then you only get fibers in some of your hemoglobin cells. And this can be advantageous. And when you get sickling of the cells, as your blood goes through the spleen, your body removes those malformed blood cells. And if you happen to get the malaria parasite, what it does is it decreases the pH of your erythrocytes, and it infects and it favors the T state. And the T state at lower pHs actually favors sickling. And so what happens is if a red blood cell is infected with the malaria parasite, then it is more, and if it has this mutated hemoglobin, it's more likely to form a sickled cell. And if it's sickled, then the spleen's more likely to remove it. So if you got a small amount of these modified hemoglobins, essentially your spleen can identify those that are infected and remove them. So people that have this actually have some resistance to malaria. Now, people that are homozygous, they're always going to be producing the mutated hemoglobin, and not all cells are going to sickle, okay, but what's found is that they tend to produce a larger level of fetal hemoglobin with a gamma unit instead of the beta subunit. And so they tend to have a milder disease, or you can take hydroxyurea, which for some reason increases that fetal hemoglobin. Anyway, really interesting that just a single mutation can cause such a dramatic change. So that ends oxygen binding and our discussion about hemoglobin and myoglobin. So we are going to go next to other case studies. We're going to discuss muscles, and we're going to discuss antibodies. Now, I think everyone is somewhat saturated with information, and there's a lot of detail in these next two sections, especially on muscles. So we're just going to try to hit the high points, try to be clear on what you need to know. There are these checkpoints here that you should know. Sorry. Yeah, so these are key concepts, and oh, I don't know why checkpoints there. Oh, I'm just a mess. Anyway, so these should be the checkpoints that we know at the end. Okay, so like I said, there's lots of structure and details when we start talking about muscle proteins, and you can just see that here. So you got a muscle, and that muscle is made up of bundles of muscle fibers. That bundle of muscle fibers is made up of myofibrils. Around those myofibrils, you have capillaries, and you have nuclei, and all sorts of things, but we want to just get down to this myofibril level. You can see in the myofibril level, we've got the sarcomere, we've got I bands, A bands, Z disc, M lines. And we've got to understand that, this isn't even down to the protein level. So our muscles have multiple levels of organization. But we're really going to just focus on what's happening at the myofibril level. So anatomy of a myofibril, like I said, there's lots of terminology here, so I tried to break it out here. We've got the I band, and so this is the light region. So if you actually took the muscles out and looked at them under a microscope, you'd see a light region. So this contains thin filaments. You see the A band, this is the dark zone here, and it contains only thick filaments. This darker zone right here is where the thick filaments and the thin filaments actually overlap, and you can kind of see that right in this case here. So there's a thin filament, here's a thick filament, and this region that is extra dark is where the thick and the thin overlap, but that's still part of the A band. Then you've got the Z disc or the Z line, and it actually cuts in half the I band. And we'll see that a little bit later, but essentially the I band kind of goes in both of these directions there. But from one Z line to another Z line, that is what's referred to as a sarcomere. That's the functional unit in a muscle fiber. So we talked about the Z disc that actually cuts in half an I band. We'll see that on the next one. And then the M disc actually kind of cuts in half the A band, and it arises from a bulge at the center of each thick filament. So it's kind of this, there's bulges here and other proteins involved that we'll talk about that are found in the middle of that, and that is what's referred to as the M disc. So I wanted that simpler picture before we came to this one here. So again, we got the picture of kind of the complete muscle fiber, and again, we're just talking about this small part of a muscle. So you've got, the muscle fiber that then you assemble several of those muscle fibers into the actual muscle, not several, many. And so if we just look at it from the side, you can see this repeating pattern. So this is actually, again, a picture of muscle. So the fibers are actually running in this direction here. These myofibrils right there. And so if you take a closer up look, you'll see this light band and you'll see the dark band. And like we talked before, this I band right here is bisected by the Z disc or the Z line. You've got this dark band that's known as the A band. And then you've got right in the middle of it what's referred to as the M disc. The lighter of the dark band is referred to as the H zone. Okay, now I'm not just saying that you need to memorize it, but what we want to do is understand kind of what each one of these visualized regions, how does it correspond to the protein organization and also function. So let's talk about the thick band. This is what's going to, the type of protein that's going to be found in those dark regions. So the thick band, about 150 angstrom filaments. They're mostly the protein known as myosin. The thin bands that we can see again over here where you get the light color, they are mostly actin. And again, this is the region where you would overlap and get the darker regions. So the thin filaments are actually bound to the Z line or the Z disc here, and going from here to here, again, is referred to as the sarcomere. That's kind of the functional unit. And why we say functional unit is what happens is you slide these filaments past each other, and so you can see that we can contract, which is what muscles do. They contract and pull those Z lines closer together. And so, in fact, muscles, when they contract, they can actually get 1/3 the length of their extended. Okay, so the model of this was proposed by Hugh Huxley, and it's called the sliding filament model. And what it is that these I and H zones decrease on contractions, but the actual thin and thick filaments stay the same length. So as we undergo contraction, this macroscopic visualization that we see actually starts to disappear, meaning that the light and dark bands start to disappear. And that is because these individual filaments are starting to slide and overlap with each other. And so you can see if there was some way that we could slide or pull these yellow pieces together, we could actually get muscle contraction. And that is what happens. So, like I said, the thick filaments are mostly myosin, and myosin consists of 6 polypeptide chains. So there's 2 heavy chains, and you can see that right down here, they actually have a coiled structure, kind of like keratin. And then they have these kind of bulky heads there. Then you got 2 pairs of different, what they refer to as light chains, and they wrap near the head there. You've got the essential light chain and the regulatory light chain. So this alpha helix here actually has a pseudo repeat, so A, B, C, D, E, F, G, much like keratin, where again, those positions at A and D tend to be non-polar, hydrophobic. So again, those hydrophobic patches are going to be able to line up with other myosin fibers there. Now, this picture off to the right is actually just essentially the myosin head right there. So you can see that it's kind of globular. And the myosin tail actually goes on for quite a ways, and they're not showing that. And then you can see these essential light chains and the regulatory light chains around there. So here is kind of a picture of a full myosin fiber. Looks like a 2-headed snake there. And if you just take the myosin fibers, what you find out under physiological conditions, they'll actually aggregate. And so their tail ends kind of face each other, and then you get these myosins, and you can see the head groups out here. So several hundred of these can aggregate. Now, what's interesting is there's an ATP binding domain on these myosin heads. It's actually an enzyme, an ATPase, that actually hydrolyzes ATP. And so there is a binding in a fairly deep pocket on those myosin heads, and that's going to be important. So again, what we do is we go from these individual myosins to this structure. So let's talk a little bit about actin. So actin is the other protein. This makes up the thin filaments. This is actually the most abundant cytosolic protein in eukaryotes. So if we divvied you up, you'd have more actin than any other protein. Now, actin has 2 forms. We refer to it as G-actin, or the globular, and this is the monomeric form. And that is the structure that you can see over here. Now, we can take this monomeric form and actually polymerize it, meaning get a quaternary structure by assembling these monomers, and we refer to that as F-actin, or fibrous actin. So G-actin actually has 4 domains. So this is a single polypeptide, but with 4 domains, and it binds ATP with calcium or magnesium in this cleft right there. And this also catalyzes ATP hydrolysis. So the myosin catalyzes ATP hydrolysis, and actin catalyzes ATP hydrolysis. So again, the G-actin, which is just the globular or the single monomer, can polymerize. And so each one of these little dots right there is representing an actin subunit. And again, they polymerized into this F-actin, which is the fibrous form. So it's hard to actually get a good X-ray crystal structure of actin. So the best one that they can get is by cryo-electron microscopy that we talked about. So it's actually a double helix, a double-stranded helix of subunits. So let me go back here. So you can see these individual subunits here. Now within each one of these subunits, you got to understand that there's alpha helices in there. So when we talk about alpha helices and then helical structure, they're 2 different things. So this is a single one of those dots. And as they polymerize, those dots actually form a helical structure. And so F-actin is a double-stranded helix of these subunits with a head-to-tail orientation. And so these F-actin fibers actually have polarity. So you actually have an end that exposes that nucleotide binding cleft, and that's referred to as the negative end. And then you've got the positive end there. Now the positive end is what binds to that Z disc. Sorry to make you motion sick, but I'm going to go back here. So this would bind to the Z end right there. And that would be the positive end of the actin fiber, and then this would be the negative end of the actin fiber. Oh, okay, that's all we wanted to say there. And now what we want to do is look at how these thick and thin filaments interact. So in kind of the shaded, less colorful part there, that is the actin fiber. And you can see kind of each one of these proteins here. And you can see that it kind of has a twist around there. And so this is just one subunit right there. And this is the myosin head right here. And so the blue part is where the ATP binds. The long myosin tail is not shown here. But there is a fairly strong interaction between myosin and actin, these two fibers here. Okay, so now we kind of hopefully better understand this picture. So here's the actin here, here's the myosin right there. These little lines here are trying to represent the myosin head interacting with the actin fiber right there. And again, the plus end down here is, again, hooked to the Z disc. Now, on the Z disc, you actually have actin running in two different directions there. So when you're bound here, you've actually got the actin. So you can actually look at the actin as being the stationary point. So essentially, they've got bidirectionality on that Z disc right there. And then the heavy fiber, the myosin, is actually going to be the mobile part. And what this mobile myosin is going to do is walk towards the Z disc. And as it walks towards the Z disc, that fiber can't really stretch, and so it pulls those Z disc towards each other. But there's many other proteins in here. Okay, so don't worry too much about these. Just understand that it's more than just actin and myosin. And so you got alpha actinin that attaches oppositely oriented thin filaments to the Z disc. So there's another protein that attaches the actin to the disc. There is the largest protein known as titin, and it actually has 300 globular domains. And so it actually spans this distance right here. And so it's thought to keep the thick filament centered on the sarcomere. And so it actually helps prevent overextension. So, you got additional proteins here and additional proteins here. There's another big protein, nebulin, associated with the thin filaments, and likely acts as a template for the actin polymerization. Remember, we got to build up the actin from individual G actin units. And this nebulin is likely a template, so it actually sets the thin filament length. You don't want it to get too long. And how you prevent it from getting too long is you're going to cap the negative end. And that's this tropomodulin right there. So it keeps the F-actin at the right length and prevents it from continually growing. And then you can also cap the actin at the Z line there and prevent that from growing. So again, just understand that there's a lot more proteins involved in there. And just here's a couple more. Myomycin. It's a myosin binding protein. So it actually binds tighten and probably helps with the thick filament assembly. So essentially to assemble these filaments, the thin and the thick, we need additional proteins to help assemble them. And dystrophin is a rod-shaped protein that associates with transmembrane glycoproteins at the inner surface. And so in the end, what that really does is it helps anchor the F-actin to the extracellular matrix. So when you condense the cell or contract the muscle, you don't want the membranes to be torn away. So there's a protein that helps anchor that and prevents these fibers from pulling on the membranes and essentially shredding the cell. And you may have heard of muscular dystrophy, and this has to relate with that dystrophin protein. It's really found in very small amounts, 0.002% of protein in healthy cells. But remember, it's important for anchoring the actin to the cell and helps prevent the cell from being degraded as muscles contract. So there's two types of muscular dystrophy. Actually, there's many subtypes of it, but you got Duchenne, I believe is how it's pronounced, which is severe, and Becker, that's less severe. And in the Duchenne, the DMD, the onset's early, between ages 2 and 5, and the muscles begin to degenerate at a rate faster than they can be regenerated. So essentially, as the muscles contract, you're starting to shred your muscle cells, and you just can't build them up as much. And then the Becker's less severe. It's onset between ages 5 and 10, and its effects occur slower. So they both arise because of mutations in this protein, the dystrophin. And in DMD, the Duchenne muscular dystrophy, it's not detectable in the muscles. So for some reason, it's not being made. Like I said, there's several different subtypes of DMD. I had a neighbor that has this genetic, anyway, I believe it's found on the, let me get this right, the X chromosome. So it's more likely to rise in males. And so there's several different subtypes, and one of them is a mutation that doesn't make full length dystrophin. I keep saying that differently. Pick your poison. But it doesn't make full length. So they've actually come up with a medication that actually will go in, and when you're making the protein, it actually makes it so that the translation from the mRNA to the protein occurs. So somehow it bridges that gap. Normally, when you're making this protein in people with this mutation, it stops and you just get short fragments that are non-functional. But if you can get kind of a read through, then you get full length. Anyway, there's some very interesting medications for this, but I think in a lot of cases, they just don't make this protein. There's like a stop codon in the middle of the gene, and so it just stops its synthesis. So I just threw a bunch of other different proteins at you, but the ones that you definitely need to know are actin and myosin. They're going to be the main contractile proteins that will be focused, and we want to understand how this works. Like I said, the thick fibers, being myosin, have this head group, and that head group will interact with the actin, which is the thin filament. Remember, the thin filament is rigged or fixed to these Z discs here. And so again, what we're going to do is get those the myosin filaments to walk along here. The thick filaments aren't going to extend, so as they walk along, they pull the Z disc closer to each other. And so let's see how that occurs. So this is in your book, and I'll just quickly walk through. But essentially, you get this interaction between the myosin head. Here's the thick filament. So this myosin head kind of sticks out of that thick filament and will interact with actin. And as we said, in this head group here, there is a ATP binding protein. So the ATP will bind to the myosin head, and it opens up this binding site. So it makes it so that the myosin head doesn't really interact with the actin anymore. And as that ATP active site binds the ATP closed, what you do is you actually cock or move the myosin head. So here you can see it kind of has an acute angle, and then it gets kind of a more obtuse angle as it binds the ATP. And so as it's in this cocked position, it will weakly interact with the actin filament again. And as it interacts, it's going to hydrolyze the ATP and release the phosphate. And as it does that, it results in strong binding of the myosin head group with the actin. And as it is hydrolyzed and it gets that strong interaction, you also get kind of a release of that previous setting of the spring here, and you get what they refer to as the power stroke. And so now that head releases, pulls the actin fiber, and then the ATP is released. And remember that myosin actin interaction is kind of weak. And then you get the ATP that makes it even weaker, and then you get recocking of the head group, hydrolysis of the ATP that causes binding and movement of the head group, and then release of ATP and kind of a weak interaction that gets weaker as the ATP binds, and you can continue the cycle. So essentially, this myosin head group is just ratcheting itself along the actin fiber and pulling those Z discs towards each other. So I don't like that they're pointing those Z discs. So the Z discs are here, and the Z discs are actually moving towards that thick fiber as the myosin head groups walk down the actin fiber. Oh, sorry, sometimes I just have the same slide. But we got different words on this. So ATP hydrolysis makes this cycle essentially irreversible. So once it grabs that actin and pulls that, you're not going to slide past. And so each myosin head groups goes through around 5 cycles per second during muscle contraction. So 5 times a second, it's going to go. Oh, did I get 5 in there? Anyway, so it's going to ratchet along there 5 cycles per second and walk down that actin fiber. Okay, now there is 2 other proteins that we probably ought to talk about that are involved in this. So besides actin and myosin, you should know tropomyosin, and it's a parallel alpha helical subunit, about 400 angstroms long, and again, another coiled coil. We start to see that kind of common. And tropomyosin subunits form head-to-tail cables that wrap around the F-actin, so the fibrous actin. And in this drawing over here, sorry, it's blue, and you can see it wrapping around and in between the myosin heads. So the F-actin is blue, and the myosin heads are orange and green, and tropomyosin is going to wrap itself around there. Actually, I think there's an error there. Sorry, the F-actin should be red. And the tropomyosin is what's blue. It's this linear chain that's wrapping around there. We'll see a better picture in a second. Sorry about that. And then you got troponin, and you got troponin C and troponin I. So they bind calcium ions. The troponin eye, which is shown blue here, resembles myosin light chain and has inhibitory segment that binds to this troponin C. Again, don't worry too much about the details, but essentially these bind calcium. And these are going to help regulate the actin-myosin interaction. So what can happen is nerve cells will release calcium, which will activate the actin-myosin interaction. And that's very important because we don't just want the myosin walking down the actin, we want to be able to control it. And so we can control that by release of calcium. And that's what these two other proteins involved in the process do. So that's the take-home message is the other ones. So you should know what actin and myosin, the role they play, and then these two other proteins are involved in the regulation, and so that we only get the walking of the myosin when we have calcium around. Okay, so this is why we need to know the tropomyosin and the troponin. So they actually modulate the access to the binding sites on actin. And how they do that is a nerve impulse will release calcium, and then that calcium will come in and flood your muscle fibers. And then these proteins here will bind the calcium and cause conformational change in the tropomyosin troponin complex. And that exposes the myosin binding sites. So these other two are there to make sure that those binding sites are only exposed when calcium's around, so that we can now control this process by release of calcium, and the release of calcium is controlled by nerve impulses. And we'll talk about that when we get to second messengers. So preparations of intact thin filaments, the F-actin, contract only in the presence of calcium. And so, like I said, it's regulated by troponin C. So we can see this process here. So nerve impulse will release calcium, and you go from 10 to the -7th concentration to 10 to the -5th concentration. So you're actually increasing the concentration twofold. The 10 micromolar calcium concentration will trigger a conformation change in the troponin. And the tropomyosin that exposes the myosin head to the binding sites. So we can see that over here in gold is the F-actin, so our polymer. Tropomyosin in low calcium is going to be red, and so that's going to be blocking binding sites for the myosin head group. When you add calcium, then you're going to see a shift here. The tropomyosin is going to move, and it kind of slides out of the way and exposes binding sites for myosin. And so just by the release of calcium and having this tropomyosin move, you increase myosin's affinity for actin by about 10,000-fold. So kind of a slick way. Essentially, you just got this coil wrapped around there, and once you bind calcium, it changes shape and exposes a site so myosin can actually get in there and bind. So something else that's interesting about actin, as I said before, it's the most abundant protein in the cytoplasm and in eukaryotic cells, like 5 to 10% of the total protein in there. And so actin can be used for other things. So you have non-muscle actin, and it can form a 70 angstrom diameter fibers, which are known as microfilaments. And these are important in determining cell shape. They help in cell division, pull your chromosomes apart, endocytosis, when a cell takes in something, and organelle transport. So we got a picture over here of a fibroblast, and it can move by disassembling and reassembling their actin filaments. And here's a video that you could watch to see that in action. But let's see if I can help explain how that occurs. So there is a question, why do I need to know the positive end and the negative end of these actin fibers? So ATP-G actin meaning when you have the G, just the monomer of actin that's bound to ATP, can bind to both ends of the actin filament. So you can kind of see that here. We got a positive end and a negative end. And so just kind of ignore the green for a second, but it can bind to this end or it can bind to this end here. But when it binds, it activates the F-actin subunits to hydrolyze ATP. So when this G-actin binds, you hydrolyze the ATP. And when the phosphate dissociates, you get a conformational change that reduces the affinity of the ADP F-actin subunit for neighboring subunits. So again, when this binds here, you're going to get hydrolysis of ATP, and then that subunit right here has less affinity for additional subunits. So the key to this is ATP hydrolysis occurs slower at the positive end than it does at the negative end. You're getting hydrolysis faster here, which means that this end has less affinity for additional monomers to come in. So you can kind of see that as one monomer comes in, several leave because we're losing affinity because the hydrolysis of ATP is occurring faster there. At this end here, you're getting more monomers to bind because the hydrolysis is occurring more slowly. And so as you get that hydrolysis, yeah, maybe only one leaves. Now these in green, that's just a marker so that we can actually watch this. And so what happens is you get more of these subunits again. They tend to bind to the positive end where you get slower hydrolysis. The hydrolysis is happening faster at the negative end. And so essentially what you're doing is this is growing in one direction. You're growing towards the positive end and the negative end is leaving. And so these guys right here, once they leave, they're going to go around and find a place and they will end up adding to the positive end. So you can see that this is growing away from that green segment there. And so this is thought to be one of the earliest mechanisms of motion. Like how amoeba can move is you just are assembling and deassembling your F-actin, but you do that in a directional method towards the positive end. And so if these fibers start aligning and they move in one direction and they push up against the edge of the cell, then that cell can start to move like you see in that picture off to the right there. Okay, that's about enough about muscles. So let's quickly talk about antibodies. And like I said, as we go, we're going to anyway, kind of limit what we need to know. So antibodies, this is how we can respond to invaders in our body. Excuse me. So our skin and mucous membranes, they're supposed to keep things outside of our body that we don't want. But sometimes things get past the skin and the mucus and membranes and they get into cells or they get to surround our cells. So they get past our first line of defense. So we have a couple other additional ways of defense besides just walls. We have cellular immunity. So these are things that eliminate virally infected cells and fight against pathogens. And this is mediated by T lymphocytes or T cells, which mature in the thymus. This is more cellular biology, so we're not going to talk much about cellular immunity. We're going to talk about humoral immunity. And humerus, why it may be a funny word, actually is an old word for fluid. So this is going to be kind of the immunity that we have in our fluids here. And these are effective against bacterial infections and extracellular phases of viral infection. So this is where we build up antibodies. So it's mediated by a collection of proteins, like I said, that are referred to as antibodies or immunoglobulins. And these antibodies are produced by B cells, beta lymphocytes. These mature in the bone marrow and they circulate through the blood. And what they have is on their surface, these immunoglobulins. And these immunoglobulins, every so often, they will run into something they have affinity for, and that is what we refer to as an immune response. So these beta cells, they have these antibodies or these immunoglobulins on their surface, and when they happen to run into something that that antibody has an affinity for, that antibody will bind. The thing that it binds to is referred to as the antigen, and then the B cell will engulf the antigen and the antibody. And when it takes in the antigen and the antibody, what it does is it doesn't completely digest the antigen. It'll actually display fragments of that antigen on the cell surface. And so what it does is essentially it's showing off, let's just use maybe not a nice analogy, but it will be, oh, well, let's think of a different one. That one's, so let's say that you're in war and you go and conquer someone, and then what you're going to do is display their hat, or let's say that they have a medal. Oh, here's a better one. Okay, dogfighting. Sorry, it took me a while to get to a politically correct analogy, but dogfighting. So the dogfight, you take down a plane, and then on the side of your plane, what you're going to do is paint a picture of the plane that you shot down. So you're going to collect, not necessarily fragments, but pictures. So these B cells, they're going to attack these antigens and engulf them, and then what they're going to do is display on their side these antigens. Well, along will come a T cell, and they will recognize these antigens there, and then what they'll do is they'll stimulate the B cells to make more antibodies. So again, in our poor analogy, you'd see a fighter pilot that has lots of different planes on his plane, and then what you're going to do is clone that pilot, because you know that pilot's really good at taking down foreign invaders. And then those new beta cells, or these new pilots, will circulate, and they will release that antibody, which will again bind the antigen. So this is a huge area called immunology, and we're not going to even really touch the surface. We're just want to focus on the protein, which is the antibody, and a little bit about the antigen complex. So this whole process, actually a lot of it's not understood. Why don't antibodies attack ourselves? How do we recognize ourselves? A lot of this really isn't understood. But most beta cells only live a few weeks unless stimulated by antigen bonding. So essentially what you do is you just send out a bunch of pilots, and these pilots don't have long lifespans, and if they don't find someone to shoot down, they feel bad about themselves and crash their plane and die. There are a few long-lived beta cells. These are the memory B cell. I keep calling them B cells. I think they're B cells. So late in the day, yeah, or sorry, I keep calling them beta cells, B cells, memory B cells, and these can recognize antigens years after the first detection of the antigen in the body. And so this is how we can actually build a response to like chickenpox, because of these long-lived B cells. So when we first get exposed to an antigen, what's going to happen is we're going to have a response. Not much of a response, but we're going to start producing a response until eventually that antigen is released. And that's what's referred to as the primary immune response. And so it tends to be weaker. And then if we happen to get exposed to the antigen again later, then what happens is we have a much greater immune response, a secondary immune response. And the reason for that is we tend to have those antibodies already built up, and they tend to be produced much faster in much larger amounts. So back to my poor pilot analogy. So under the first attack, we have only got a few pilots around, and they mount an assault, and they beat off those antigens. And then they paint on the side of their plane, the type of planes that they shot down. And then they just kind of go dormant. A lot of them just kind of die out. But there's a few that sit around and reminisce about the good old days. And then when they're attacked again, they say, "Ooh, I know how to beat that invader." And they'll take that pilot and clone it. And then you'll have a bunch of these cloned ones, and then they can mount a much faster and more vicious attack against the invader. But you can see if you're introduced to a different antigen, then you just get the primary response again. So the secondary response is much faster because you have these memory B cells that are floating around that are already have the antibody within them. And anyway, once they recognize the antigen, there's a process where they'll be replicated and you'll produce lots of those B cells. So again, that's really immunology that we just poorly touched the surface on. But since this is a protein chapter, we want to focus on the thing doing the action, and that's the antibody. So antibodies have conserved and variable regions, all contain at least 4 subunits, and 2 of those subunits are identical and referred to as light chains, and those are what are colored in magenta. And then you have 2 identical heavy chains. Those are, in this case, yellow and cyan. And the subunits of an antibody associate by disulfide bonds, and we'll show you those. And they end up in kind of this Y conformation. So that's why I had this picture here, and you can see that it kind of has a Y conformation. And so the light chains are going to be found up here, and the heavy chains are going to extend all the way down. So there's classes of these immunoglobulins or these antibodies. Again, if you were taking an immunology class, we'd spend a lot more time talking about them. And I'll just let you read. You don't need to know these, just understand that we have different ways or different types of antibodies that essentially recognize different things. But what we really want to get into is how do these antibodies recognize other things? And so they can digest a protein with papain, which is a proteolytic enzyme, and it actually cleaves here, and it cleaves it into 2 different groups, what they refer to as the Fab fragment, which is the F antigen binding. So they knew that this was the part that bound the antigen, and so it cleaves here, and you get this part that binds antigens, and then you get the FC fragment, which is the easy to crystallize fragment, which is kind of this stem down here. And then within this, so those are kind of what, when you talk about the Fab and the FC, so the fragments, that let them know that the recognition of the molecule is happening up at this end. Now, we said that it's composed of 2 identical light chains, and here's the light chains here. And then the heavy chains, like I said, extend down. Here's the N-terminus of each of those, the N-terminus of the light chain and the N-terminus of the heavy chain. And this is what's referred to as the variable region or the antigen binding region. And then you have some conserved regions in the chain. So this is the conserved region in the light chain. And then you've got a couple conserved regions in the heavy chains. And you can see that where you're getting the disulfides here. So you get disulfides within the heavy chain and between the heavy chain and the light chain and between heavy chains. So essentially this whole system here is all cross-linked into kind of one unit. And what you find out is that there's actually lots of kind of repeating units here. And so it's thought that these antibodies actually came from duplication of a single gene 6 times. There is variability, but there's some conservation of the sequence there. Okay, but really at those ends there, you have a lot of variability in the group. So this, I believe, is just the light chain. And so again, in the light chain, you had the variable region and the constant region. And these have shapes. But in the variable region, they even refer to it as a hypervariable region, you've got lots of different loops there and lots of different amino acid sequences that can be found here. And then this is going to be kind of a constant domain with a constant structure here there. So you get these 3 variable loops, and this is where you really get most of the variability in the antibody, in those loops right there. And so you can kind of see that. You get these variability in these loops. And so really, you get a bunch of different sequences here. And every so often, you just happen to get a right sequence of amino acids that has a complementary electrostatic or hydrophobic interaction with the antigen on the invader there. And so again, these interactions can either be van der Waals forces, hydrophobic, hydrogen bonding, or ionic interactions, but they're just variable. Really what it is just luck of the draw. And the interaction between these variable regions and the antigen can actually be fairly strong. So they're saying the KD here from 10 to the -4th to 10 to the -10th. I think they've even got some of these things down into the picomolar, down to 10 to the -14th. So some really high affinity bonding, essentially kind of a covalent type of interaction possibility. So you want this to bind the antigen fairly tightly. So most of the antibodies will bind in a divalent manner because you've got these 2 regions out here, and so you can actually bind the antigen twice. And so what this does is allows the antibodies to cross-link the antigen molecules. And so you can see here that you actually can start to form an extended network of these molecules. And when you do this, that's going to actually trigger the B cell proliferation. So when you start to see aggregates of all these antibodies and antigens, essentially what they say is, oh, we caught something, we need to make more B cells, more pilots to go out and attack. So the antibody diversity and the immune response actually comes by some recombination of the DNA. Again, we really haven't talked about how the DNA turns into the RNA and then eventually the protein. So we'll just suffice it that there's a lot of shuffling of these regions here. And so in these variable regions, you can get these groups that kind of shuffle back and forth, and you can end up with multiple different arrangements. In fact, somewhere, I can't find it, but you can actually end up with 10 to the 18th possible different combinations at those variable regions of antibodies. So most humans or organisms aren't going to nearly have that many. Oh, here it is right here. There you go. So the potential to make 10 to the 18 unique antibodies, that's a huge number. We're not going to make that. We're only going to make a small fraction of those. And again, really what it is the antibodies aren't specific for a pre-known antigen. It just happens that when you're exposed to an antigen, there's antibodies floating around, and we have so many different possible combinations that by chance, one of those antibodies will bind that antigen. And we can see that now with COVID. You know, COVID is taking hold because no one was really exposed to this virus before, and so no one has really built up antibodies towards the antigens on COVID. And it's thought that once you're exposed to COVID, you now have the antibodies, and so you're not going to get it again. Now, why you can get the flu every year is the flu mutates much, much faster, and so each year the flu virus has different groups on it, different antigens. Now, they do make a flu shot, and they try to predict which antigens will be on that flu. And sometimes they get it right, and the flu shot works, and sometimes they miss, and the flu shot doesn't work. But really, this is just kind of a hit or miss. But there is some feedback. If you get an antibody that's just random, that just happens to bind an antigen, then there is a feedback loop that says, hey, this is working out, let's make more of these antibodies. And so the type of antibodies that we have in our body when we make them are what are referred to as polyclonal antibodies. So what a polyclonal antibody is it's a mix of antibodies that bind to a specific antigen. So if you actually went and looked at them, you'd see a lot of variability among those antibodies, but they all have some affinity for that antigen. Now, what would be really nice is if you had monoclonal antibodies. And what a monoclonal antibody is really a single molecular entity, meaning the sequence among all the antibodies is going to be identical. And so antibodies are very useful. We talked about them a little bit when we're talking about protein affinity chromatography. I believe we'll see them later on. But you don't really want antibodies that have variable affinity for the antigen. I mean, it works for us, but if you're doing experiments, you want to know what type of antibody you have in there and what affinity it has for the antigen. And so they've come up with ways to make monoclonal antibodies. Now, normally a single B cell isn't immortal, meaning that it's just not going to keep replicating and producing that antibody. So somehow we need to make the B cell, we need to make it immortal, meaning that it will replicate forever and never die out. And so what they did is a trick. They took some mutant mouse myeloma cells. These are cells that we refer to as being immortal, meaning that they can replicate forever. And then you took some mouse lymphocytes from the spleen to make an antibody. And so these cells right here can only grow if you have, I believe, purines in there, purine bases. They lack this machinery to make them. Now, these have the machinery to make the purines for your DNA. And so what happens is you combine these two together, and those cells that actually join or form a hybrid or hybridomas, those right here are now immortal. So you have a mouse lymphocyte cell, and then what you do is you test these immortal cells for antibodies towards your target of interest. And then you find those that have antibodies towards your target. And in here, you've got an immortalized cell that will replicate forever and only produce a single type of antibody. And when I say type, I mean really a single molecular entity, a single sequence or single protein, meaning it doesn't have variation in it. And so these can actually be used as therapeutic agents. So here is one. And actually, if you ever see drugs that end in AB, they are likely antibody drugs. So they're actually these antibodies that tend to bind specific targets. So in this case here, this will bind interleukin-6 receptors to suppress the immune response in patients with diseases like rheumatoid arthritis. So you'll actually take an antibody, it binds a specific protein and shuts down a disease pathway. There's some other ones that the book talks about. You may have heard of Herceptin. I believe that's its name. It actually binds to the HER receptor, which is found in, I believe, 25% of all breast cancer patients. And so what it does is it binds to that receptor. Those breast cancer cells don't grow anymore, and they may even shrink, but the antibody essentially will bind. These are very expensive drugs because they're what are referred to as biologics. They're actually protein drugs that have to be very clean. And again, you want monoclonal antibodies. You want to know that it's going to bind its target with high affinity. And so sometimes we actually get diseases, or sorry, immune responses against ourselves, and those are referred to as autoimmune disorders. And it seems like there's actually a lot of these that are occurring more and more. And there's thoughts on why that may be, but celiac disease is one of them. Lupus is another one, and they're just these autoimmune disorders where essentially our immune system is attacking our own cells. It's for some reason recognizing those as foreign and is making antibodies. And when those antibodies bind molecules within our body, if they're invaders, that's a good thing. If there are normal functioning cells, that can be a bad thing. In fact, it's thought in some cases of like diabetes, the antibodies are attacking the islet cells in the pancreas and essentially killing them, and so you don't make insulin anymore. Anyway, there's a whole type of these, even rheumatoid arthritis and connective disease. I had an issue one time that was interesting. I actually had what are referred to as anti-nuclear antibodies. These are antibodies that recognize the phosphate in the DNA. It's kind of an indication of rheumatoid arthritis. It disappeared. I don't know why, but at the time I was having lots of joint pain and stuff. So my antibodies decided that they didn't like my DNA, and anyway, started attacking themselves. But lucky for me, it kind of went away. I'll just let you read this. I think that is enough. I think I've been talking for over an hour, if you can't tell by my tongue-tiedness. But the key for these muscles, make sure that you understand actin, myosin, and how you can regulate actin binding to myosin, those proteins. Don't worry about all the other ones. And then antibodies, understand what they're good for recognizing foreign material, and just generally, how does that arise? It arises because we put these variable regions at the end of the antibodies. Our body has ways to really shuffle the types and types of amino acids in those variable regions. And we just make lots of these antibodies, and sometimes they just happen to recognize an antigen. And then there's a feedback loop when they do recognize that antigen that we produce more of those antibodies. And that is it for this section. That is a slog of information, and I will hopefully be posting a test study guide. And that's why I moved the test back to Friday, because I want to give you at least a day to work through that test study guide. So I'm going to write a test and then kind of give you a hint of the important stuff to know for the test, because this was a bunch of information.